Thank you all for joining us today. I just wanted to give you a little background about today's special speaker. This is Dr. Theresa Boyle. Theresa Boyle practices as a molecular pathologist and a lung cancer clinical investigator at the Moffitt Cancer Center and Research Institute and University of South Florida. She received her pathology training at the University of Colorado and Stanford University and is board certified in anatomic, clinical, and molecular genetic pathology. Her clinical duties include interpretation of molecular genetic results for patient care, service as a medical advisor to the personalized medicine group, and as lead pathologist for several clinical trials. Her research focuses on biomarkers of lung cancer. Dr. Boyle, thank you so much for joining us today. We're so looking forward to your presentation.
Well, Dusty, thank you so much for the kind introduction. And I also really appreciate the invitation to speak here. Thanks for thinking of me, Ed. We've had a few interactions now, and it's always nice to be able to talk with people in the community. And I'm particularly excited to talk about the rapid tissue donation program. It's really my main project, and I just really enjoy talking about it. So I'm happy today to share it with you. So let's go ahead and get started. This picture here is actually a picture of tissue that we collected with the thoracic or lung cancer rapid tissue donation program at Moffitt. And you can see in the light blue aqua color that these are the cancer cells, and it's an immunofluorescent assay. And then this colorful kind of light bright looking background is the stroma that's inside the tissue, and the cancer cells are invading into the tissue. And I think the little blue dots represent the nucleus of the cells, and the yellow and the green and the and the red are different markers. We can look at many different types of cells with these fluorescent markers by just tagging whatever is on the surface of specific cells. But we can't do anything like this without the tissue. And having tissue for research in lung cancers, it's a big hurdle for the researchers. There just isn't much tissue available for the researchers to learn more about the cancer and how to provide better patient care in the future. So we're very, very grateful to the participants in our program who make this all possible. This program was started back in 2013 by Dr. Eric Hora and Gwen Quinn, Matt Shabath, Teresita, Jessica, and Christy Pratt. And there's a lot of coordination of the tissue collection with the Tissue Corps, and Michelle Fournier has helped with that.
Yeah.
So to start a program like this, there's obviously quite a few ethical issues that first need to be addressed. And so we're very fortunate to have Gwen Quinn, who's really devoted her career to studying ethical issues and epidemiology. And the first question is, how do you even approach and consent patients with cancer into a program like this? And then to study what are the barriers, what are the things that help, or the facilitators for setting up such a program. And then after you've collected tissue, what are the desires of the next of kin for future communication? How do you show your appreciation for the participation? And so the studies of the ethics began in 2013, and the program didn't actually start until 2015. And it took a couple of years to sort through some of these ethical issues. And the program was initially started with the goal to recruit 20 people with advanced stage lung cancer. And let's see if I can move this out of the way too. Yeah. So in the beginning, it started with conversations, and that this is an example of one conversation of Dr. Horror with Gail Porter, one of the first participants in the program. And he said, I need your help. And he said, we have this new program, this new research program, but I don't know how to present it to a patient because I don't want to take away their hope. You know, when a patient comes to the hospital, the goal of the oncologist is to take care of the patient, cure the patient, get them better, and send them off to enjoy their lives. And so how do you bring up the topic? Well, we have this program where after death, it would be really helpful to be able to collect your cancer tissue. It's a very, at least for the oncologists, it's a very sensitive subject that they don't feel comfortable bringing up. So they needed help.
Would you rather us hold our questions or?
No, feel free to interrupt and ask questions on the way.
Because I'm fascinated with this topic, but I didn't realize that the tissue donation is after death, because there are some programs that you donate your tissue, your tumor tissue, you know, when you're still in the trenches fighting.
True.
So if you could maybe just give us a little bit more information. So it's only after death. You don't need that. You're not for this program using anything until the patient dies. That's
true. Although we are thinking about changing the protocol to allow blood collection for the program. And after death, we do have IRB approval to also do research with tissue that was collected during clinical care, but we're not using the tissue for research during the clinical care. It's only after death that this program would access that tissue. And the reason is during clinical care that there's not much tissue, and there's just, and it's needed for most of it for clinical care, for clinical trials, to take care of the patient, and research is lower priority than that.
And it's not easy to access.
And it is not easy to access. Yeah. And that's just been, you know, a chronic problem for lung cancer researchers for, you know, all years since I've had my career. It's like the tissue is the issue, is the saying we have. Yeah, so good question. And everybody feel free to ask questions on the road. I'm not feeling like I have to get through the slides quickly. So anyway, she and her family helped. So this is a picture that this is Gail Porter right here, and her daughter Celia, and her granddaughter Riley, and she really helped get this program off the ground, and she helped Dr. Hora come up with ways to communicate about the program in a way that does not take away hope, because really, if you think about it, when you go and get your driver's license, you're asked whether you're willing to be an organ donor, and it's not a big deal to say yes or no. And in the case with cancer, it's a little different. You can no longer be an organ donor in the usual way, but you can donate your cancer tissue to research. But it's
well. I just wanted to. I'm sorry. I just want to interject a brief comment, because what you just said is it's just so profound, and such a beautiful way of looking at this, and it's reminiscent of a recent thing that happened in my granddaughter's life. Her boyfriend, he just turned 18 in September. And he, when he turned 18, he stood in line for like a very long time so that he could become an organ donor. And little did anyone know, just a few weeks later, he would pass unexpectedly.
Oh.
But the family had that comfort to knowing that he really wanted to do that. He believed in that. And like you said, when you get cancer, you know, you're limited on what you can donate. Maybe your eyes, but that is just,
you can donate your eyes. Yes.
Thank you.
Yeah. and this is just so valuable. But it is altruistic. Like it's not going to help. Participating does not help the person who's participating unless it makes them feel good that they might be helping future generations, future people. It's very altruistic, and I've just been so impressed by how altruistic people really are. You hear so much in the news about negative news, and this is where real life people are really trying to help each other. And so yeah, so just a beautiful family. And so she was one of the first participants in the program, and her daughter, after her mother was diagnosed, Celia, she started doing this annual Fight for Air Climb with the American Lung Association. And she would do that every year to raise funding for lung research. And after her mother passed, it was very close to the Fight for Air Climb, but she did it anyway. And after that, we did collect the tissue for the program, and we did do, we have been able to do this amazing research with it. And Moffitt now has a Gail Porter Memorial Award that we give to the top fundraiser at the annual Fight for Air Climb. So here's me. In the very first year, it was Riley, Gail's granddaughter, who rose. She was the top fundraiser for that year, and so she got the award, which was very fitting. And that this is the second award winner. So it's really a team effort. There's no other way to say it. So with this program, we hope that it provides hope in the fight against cancer, and it's a pretty simple program. With the participation, we're collecting the samples after death and doing many kinds of research. And here we talk about how this is another option to donate after death other than organ donation, and that it does not cost the family any money. We pay for the transportation expenses, of course. And it's really that the research is controlled by the institutional review boards, and it's all above board. And one support for this program has been the Latte Group. I don't know if you guys are familiar with that, the Lung and Thoracic Tumor Education Group at Moffitt. And so they've helped us create this flyer, and they help with giving us feedback about how to maintain the program and keep it going. They help with referrals. And our coordinator, Gina, is really just awesome. She's been so wonderful to work with. She helps me in every way. She's very organized, and I'm not so organized. So she's wonderful to team up with. And so I think her coordination of the program has really transcended the program into a very successful program. And so this is just some of the wording we came up with. Would you be interested in contributing to research to help others? We don't approach patients unless the oncologist says that it's okay, that it's not a bad time. I mean, maybe every time is a bad time, but preferably at when there's been some good news. And I'm certainly open to feedback from this group too about what's the best way to communicate about this program. And also, even though people with cancer cannot donate organs in the usual way, cancer tissue provides another way to help others. Would you be interested in learning more? And so if we have consent, then that allows Gina, our coordinator, to basically look at the electronic medical record and follow how the patient's doing. And if the patient's not doing as well, then she'll start communicating with her providers and and just follow up. If a patient's referred to hospice, then she'll communicate with the hospice care team. She tries not to bother the patients too much after that initial consent, so kind of working behind the scenes. And then if the death occurs, then we're contacted, and we collect the donor and collect the tissue, and we try to collect it rapidly so it doesn't interfere with any proceedings and get the transportation of the body back to wherever the family wants. So that's the hard part. There's been, after this initial hesitance in terms of setting up the program, should we even set up a program at Moffitt? How do we communicate? We had the launch in 2015 and the first tissue donation in 2016. And then we started having some publications to learn more about lung cancer. And in 2020, we began a collaboration with Bristol Myers Squibb's BMS. And so that helps fund for our coordinator salary, some of my support. We have a radiologist on the team, an oncologist on the team, epidemiologist, a statistician, bioinformatics, because we have a lot of information and data now. And we put information in the REDCap database, what medicines the patient was on, what genetic changes were there in the tissue. This is all really important for the research to be fruitful. And one thing people have always wondered about is whether the tissue collected after death is as good as the tissue collected during life. And I'm showing here some pictures of the tissue from under the microscope. So I'm a pathologist. I have to show some pictures of the tissue under the microscope. And so here I'm showing a picture of the kidney with the glomerulus, which filters the fluid in the blood and transforms it into urine. And so this is a really nice looking glomerulus. But you can see over here are the cancer cells that are kind of invading into the kidney. But this is very high quality tissue when it's collected rapidly like this. If it's not collected rapidly, you don't see these nice glomeruli in the kidney. And here in the pancreas, likewise, you don't see these nice pancreatic islets if the tissue is not high quality. So we collect the tumor tissue, we collect paired non-tumor, the primary cancer. Sometimes there's no cancer in the lung, but there might be a metastasis in the liver. And
Dr. Boyle,
yeah.
I have a couple of questions.
Please.
So you're saying rapid, but could you explain what that means, rapid?
So rapid can be a relative word. And are we talking rapid, the time from after death to tissue collection, or how quickly are we collecting the tissue when we collect? And it's really both. And people don't really fully understand what are the factors that cause the tissue to be high quality or lower quality. But we aim to collect the tissue within 48 hours after death, but our average is about 10 hours, and we've collected in 3.5, 4 hours. We've actually gotten better as we've gained experience over time with coordinating everything and collecting faster. So another change that has helped with our rapidness has been, we used to have a medical examiner collect the tissue out in the community. We thought that would be faster, but she didn't really, she wasn't doing research. She didn't care as much as, say, I do. And I'm on both sides, like I'm collecting the tissue and I'm doing research. And so, like for me, I'm kind of willing to drop things and collect the tissue as soon as possible. And Gina is great at coordinating. So we actually brought the collections to occur at Moffitt, where we have all the facilities, and we can collect the fresh tissue and get it to the research lab right away. And so that's been working better for us. But even there's publications even on, how do you assess the quality of the tissue? And one way that we've assessed that is by doing research with it. And, can we extract DNA and RNA? Can we do sequencing with the DNA and RNA? Can we do immunohistochemistry? And also, what do these H&E slides look like? And we're finding that we can do just about anything with this tissue. We're getting really good data and results from this tissue. And so we didn't know that ahead of time.
Another question was, and you might have already answered this, but do you have a certain number of tissue samples that you're hoping for? Or is this just ongoing? And one other question, you're talking about maybe harvesting at Moffitt, but what about people who, like, we've got people on this call who are in New York, North Carolina, in other locations. How, if somebody, and you're probably going to get to that, I apologize, but
That's okay.
How can somebody donate to this program?
Right. So I can answer those. I'll answer the first question first. So what's our aim for the number of tissue samples ongoing? We've been thinking about it more in terms of people, like how many people do we want to consent to the program? And it started as with a goal of 20, and then we increased it to 60, and I think it's now at 80. And we don't succeed in collecting tissue from every person. Sometimes we don't know if a death has occurred. So, but when we are notified, we usually are successful. Our numbers have increased. I personally think of this as an ongoing, enduring program without a clear end, because therapies change and you always want to know, like, what happened afterwards. And so as long as we have the participation and the funding, I don't see an end to it. But right now, what we have approved is 80 participants. We have collected tissue from 33 donors. And I have some of the numbers of participants on another slide. And then to answer the other question, the different states, we now, for inclusion, we collect from people, I think, within about 100 miles or 2 hours of Moffitt, because that's all we have a transportation company that we have a contract with, and we do want to keep it rapid. But there are other ways to donate if you live in different states, and we do have some snowbirders here in Florida. People will travel back and forth, and maybe they'll be here, maybe they won't, but that they still can participate if there's a chance they'll be here. And I don't know the names of the other organizations, but my coordinator, Gina, she does. So if you call the number, even if you're from a different state, she will provide you with information about other options.
Thank you.
Other programs, yeah. And here, I with this picture, I just wanted to highlight that here's the cancer tissue, and you see on the billboards on the highway even these days how immunotherapy, anti-PD-L1 is a fairly newer therapy that has really transformed cancer care. And here you can actually see these little purple dots are lymphocytes and you can see them attacking the cancer cells. And it's really nice to be able to see that in a real way. And one thing that prevents these lymphocytes or the immune system from attacking the cancer is this PD-L1 immune checkpoint. And so the researchers have figured this out. And now a therapeutic option is anti-PD-L1 to allow these immune cells to see and attack the cancer cells. And so it's really from looking at tissue like this and staining it for different markers that you learn about this, like what's important, what's happening, why is a therapy working, or why is a therapy not working, or why was it working for a while and then it stopped working. And so that's why it's been worth all this effort to try to figure it out and develop this program. Having this tissue has enabled studies that just weren't possible before. It allows us to study, this is a big word, tumor heterogeneity. And all it means is differences between tumor in different locations in the body. So is it different is the tumor in the lung different than the tumor in the liver or the tumor in the kidney? And it may be that the therapy worked really well with the tumor in the lung, but didn't work so well in the liver. And we need to understand why for the patient. We need to be able to help. And also maybe a drug works and then stops working, and we need to know why. And we can study the mutations, the changes in the genetics to see why, and then come up with other ideas for therapy or try to prevent that evolution that causes the therapies to no longer work. We can look at the things inside the cell like DNA and RNA. And we can also look at things on the surface of the cells, like the proteins. We hear about eating protein, but there's also protein in your body that builds your body. And we can study these things with all these different methods. And some of these require a lot of tissue, and you just don't get a lot of tissue during clinical care.
I've got a couple more questions that just
all right, cool. This is great.
So, and first, hi, Stephanie. She sent these in. So in reference to younger parents, how do we know if our tissue is in any way linked to our children to use? Maybe is there any way for this to be donated to family first, so to speak? And then I'm going to go ahead and read a couple of other questions too. And I can read them back again if you need me to. Sure. When is it the quote right time to be a part of the donating and no more donating to research within your own geometric changes? And when are the climbs usually held? That's the third question.
When are the what held?
The climbs that I think it's the ALA climbs they're talking about that you would.
The climbs. Yeah. The Fight for Air climb was last month. Steve Riddle is the one from the American Lung Association who organized it. And I could definitely provide more information.
That's fine.
And then
we sometimes will share that information, but that's another nonprofit. That's American Lung Association. They do good work. But so tissue question. Stephanie wanted to know about, because there are some.
How do we know that
genetic questions and what can be beneficial to the children of parents who had lung cancer?
Yeah, well, that's an excellent question. So now you're talking about not just the mutations in the cancer, but what we call germline or inherited mutations. And so the thing is if we collect the tissue, some of the tissue is going to the tissue core for research, and some of the tissue is going to the pathology department. And the tissue that goes to the pathology department is really intended for the next of kin or the family. And so if there's a question that the family has even many, many years later, even 10 years later, they can contact Moffitt and they can ask for that tissue to be tested. Because who knows, like 10 years from now, whether there's going to be some genetic change that might be of interest to the children. If you don't collect any tissue at all, then there's just nothing for anything. Blood samples are a good idea too for the family. If you want to make sure that future generations within that family have the tissue available to them. But we really can have that available. Now, the goal of the rapid tissue donation program, like we don't have any specific research that's looking for the germline changes or the inherited changes. But when we do our sequencing, we get both the genetics of the tumor and the inherited changes. And so we are generating that data. And but it would require communication to be able to provide that data to the family. I'm not sure how the consent is set up for that. I would think that, like, if 10 years down the line, the kids wanted to know something, that contacting Moffitt and the pathology department would be the best way. And that way it could be tested with whatever the most modern genetic sequencing platform is at that time. But we collect plenty of tissue, so that is entirely possible. And it would be through the path department. We haven't had any such requests yet, but it's a good question. I haven't even thought about that one before. Then the question, when is the right time? Really, any time is fine. I mean, just like when you get your driver's license, like it's not like somebody said that that's the right time to decide whether you're going to donate your organs. It's just that's how it's been set up. So really, any time that that a person with lung cancer decides that they want to participate is a good time.
So, Dr. Boyle, you mentioned that sometimes somebody had signed up for the program to donate their tissue upon death, and then they passed away without Moffitt knowing about it or being notified. And yeah. I'm just wondering about, does Moffitt have like a form that people would keep with their will and other
yes.
Or what is the process how does a loved one know that Moffitt needs to be contacted?
So Gina provides the patient and their family with some paperwork to try to help remind them. But, this is not the top thing on people's minds at the time of death. And so it's really a combination. For some families it is, because it might be like your story earlier, it might be like that's like really what that person wants, and the family knows it, and the family wants to honor it. And so then we will know, because that there's a lot of communication going on. But there's other situations where maybe it's just been forgotten about it. It's been many years. And so there we only are knowing because the coordinator is checking the medical chart each week. But if the patient hasn't pursued further care at Moffitt, then we just might not know unless we get a phone call. And okay. So it's really a phone call.
So thank you.
Yeah. Okay. So we have 69 people consented now, and we've collected tissue in 33, and I guess we talked, and it's fine for people to withdraw consent at any time. We don't ask questions. It's just some people, they think about it a little bit more and decide, no, I'm not comfortable, and that's fine. We just appreciate that it was considered in the first place. And this shows some information about this. NSCLC stands for non-small cell lung cancer. And so this is adenocarcinoma and squamous cell carcinoma. And the adenocarcinoma, there's often a driver gene mutation. So now I'm starting to get into some of the science. Sometimes it's a KRAS gene, sometimes it's an EGFR gene, sometimes it's an ALK fusion, sometimes MET exon 14 skipping. And so all of these are different drivers, but they're known, and there's known FDA approved therapy for at least the EGFR and the ALK changes. And for KRAS G12C, there's been a very recent therapy, sotorasib, that's been FDA approved. And then there's the different therapies. And here, you were asking about the time to collection. This shows, some in 4 hours, 5 hours. But even when it's been a longer amount of time, we've seen high quality tissue that we can do research with. And we collect a variable number of specimens depending on how many metastatic lesions there are.
Dr. Boyle, do you not collect small cell?
So we do collect small cell. Good question. So SCLC is for small cell lung cancer. And so we've collected for small cell as well. I just couldn't fit them all on to one slide anymore. I used to have them all together. We have a lot of researchers with very strong interest in small cell at Moffitt. It's not as common. Small cell is not as common as the non-small cell, but the therapies have not worked as well. Like the chemotherapy works initially. And so there's a lot more that we need to learn about the small cell. And so it's been important to also collect small cell, and it gets neglected, and
it does.
Don't want to neglect it.
We are actually starting a small cell virtual support group this month. So we're real excited about that. And if you, maybe you can share with that group at some point, but also it would be great if you mentioned some of your colleagues who are particularly focused on researching small cell. Perhaps they could also speak to our virtual small cell group in the future.
Yeah, that would be fantastic. I can give you some names. So
that would be great.
Some of the names are here: Brad Perez, Jose Canajo Garcia, and Ben Creelan is another one. But for a senior member, Alberto Chiappori. Brad's always presenting. He's very enthusiastic and energetic. But I think any of them would.
Great, and we'll reach out and get contact information from you later.
Okay, sure. Sure. Yeah. And if you need me to stop at any point, I can. But I enjoy this being more interactive. We have several different research projects in progress and more planned. I'm not going to talk about the specific research. I just wanted to show a smattering of just of the research that's being done, and that we're not just putting the tissue away into a corner and forgetting about it. We're doing, like, these are all the people involved, and this tissue is just so valuable for us trying to better understand cancer. We've been organizing the tissue into tissue microarrays. I'll show a picture on the next slide, and performing that multiplex immunofluorescence that was on the cover slide with the different markers, different colors. We've been doing some sophisticated sequencing to know the sequence inside of the tumor cells, protein exploration. With mass spectrometry, we can study like 100, even 200 proteins with a small amount of tissue and see how the proteins are different in different cancer sites. And most recently, Moffitt acquired this geomics platform that can look spatially at the different markers plus do sequencing of different areas in the tumor. It's a very sophisticated technology, and we've been using it with this tissue. And this shows the tissue microarray, and it's got each of these little dots is a different cancer sample. And so it's actually, a hole puncher, like you take a hole punch through a piece of paper. We can take, since we have so much tissue, we can take like a punch through the tissue and then put each punch of tissue into a wax block. And so it's all in one matte wax block. And then we can stain that one wax block and look under the microscope and learn so much about all these different cancer types with not too much funding. And I'll show you a picture here, that this is what we can do. Like, this is an example of like one of those punches of tissue, and how we can stain it with the multiplex immunofluorescence. And so we're here again, the aqua blue, the pancytokeratin, that's the cancer. And then CD3 are the lymphocytes, or the immune cells trying to attack the cancer. And this researcher, Amar Beg, he's really interested in dendritic cells, these DC dendritic cells. And so he's got 3 markers for the dendritic cells. And it's very, very focused research that he's doing. And it leads to these clinical trials and new therapies. And so, so this is just one of 4 panels that we're running on these tissue microarrays. And we have 6 of these tissue microarrays. So I think you can get a glimpse of how much we can learn from this tissue, and that it makes it worth all this effort to team up with people like you to arrange to collect the tissue so we can do the research and learn about the cancer and try to fight this terrible disease. And these are some of the publications. They're freely available on PubMed if anybody wants to learn in more detail about some of what we've learned. We've presented at meetings.
I've got another question. Let's see, maybe, I'm sorry, I'm trying to understand it. How does this compare, and it might have been the last slide, how does this compare to biomarker testing cancer patients biomarker testing cancer patients are now having, and while they're still alive? And then have pathologists ever found matching tissue, another cancer table together? Let's see, have pathologists ever found matching tissue, another cancer table together? Not exactly sure. Stephanie, if you can maybe clarify a little bit more, but at any rate.
I can answer that first question while you've clarified that one. I'm not sure.
Sorry, there are some typos in there, but we're trying to work it out.
For the biomarker testing question, how does the testing that we're doing on the rapid tissue donation tissue compare to current clinical testing? And I'm going to say it's we do the same clinical testing and a whole lot more. So we have this assay that I validated here, STAR NGS. It's a 170 gene panel that we run for clinical care to identify biomarkers for targeted therapy. And so I ran this very same assay on the tissue we collected from the rapid tissue donation program. And we just submitted, we're actually, by the end of the month, we're going to submit an abstract for the Association for Molecular Pathology meeting based on the results of that assay. So we have like the before testing during clinical care and the after testing with the RTD tissue. But this multiplex immunofluorescence, this is not done as part of clinical care. It's too costs too much, it takes too long. I mean, insurance would never pay for something like this. And we're learning in so much more detail and depth about the cancer with this. So
Thank you.
yeah, these are all really good questions. I really appreciate them. And I really appreciate all the participants, the donors, their caregivers. Oftentimes when we bring up the program, we'll have patients who are very enthusiastic and the caregivers are less enthusiastic. They really aren't so interested. But then we find that when we're contacted to do the collection of the tissue, that oftentimes the caregivers have become enthusiastic, that they really want to honor their loved one, and they make sure that their wishes are taken care of. And from our end, we're just very grateful because this is not this is not the usual kind of gift or donation. You know, that these are good too, but it really comes from the heart, and it's really to help against the bigger fight against this disease, and just would not be possible otherwise. So, and that really is the end of my presentation. I also want to thank all the people who are part of the rapid tissue donation team. I've talked about Gina a lot, and the people who started the program. We also have Bruna, an oncologist, who's a co-principal investigator on the protocol, and Amar Beg, who's the chief scientist driving the research, and then all the people that started the program and are doing research with it. Dr. McCauley is the pathologist who really helped us bring the collections in-house to Moffitt. He is the director of the autopsy services here. And Brad and Jose Canajo Garcia are doing some amazing things with the fresh tissue. And the funding is, of course, important too.
Absolutely.
We can't do the fancy stuff without that.